Welcome everybody and good morning, and thank you to attend to our Marine for Health webinar. And the subject today is about the potential health effect from marine ingredients on gut microbiota. So my name is Rachel Durand and I am the project manager of Marine for Health for the NCE Blue Legacy, which is a cluster for innovation and value creation based on marine resources. The aim of the cluster are to promote sustainable innovation and profitable production of marine ingredients from various marine raw materials. The Marine for Health project purpose is to establish knowledge and scientific expertise for the health effect from marine resources, and also to support the commercialization of new products. So just a reminder before to start the webinar, this webinar will be recorded, so you will have access to the video after on the webpage of Marine for Health. You will also have access to the PDF presentation from our speakers on the website also. You are able also to ask some questions to our speakers, so you can use the chat or the Q&A box, and our speaker will answer the question at the end of the webinar. So now we are going to start the webinar with a co-presentation by the Dr. Ird Musk and the Dr. Tonia Hegetset. She is a senior research scientist at SINTEF Industry at the Department of Biotechnology and Nanomedicine. She has an extensive experience within material science for medical application, including drug formulation, encapsulation and control release, physical, chemical, and biological characterization. She actually leads two SINTEF strategic focus area, which is nanomedicine and the gut microbiota. We also have the Dr. Tonia Hegetset, and she is a research scientist at SINTEF Industry, in the Department of Biotechnology and Nanomedicine. She is responsible for the sequencing facility of sequencing-based bioinformatics at SINTEF, and as in the latter years, she had a high emphasis on genomics, transcriptomics, and metagenomics, including shotgun, barcode sequencing study, as well as nanopore sequencing. So now, Ird and Tonia, the stage is yours.
Thank you very much, Rachel, for the introduction and for the invite to talk today about microbiota activities at SINTEF. But first, a little bit about us. So SINTEF is, as many of you probably know, one of Europe's largest independent research organizations. We're around 2,000 employees. It's a private, non-for-profit research institution. And we work across many different disciplinaries, from ocean to space. So Tonia and I, we are a part of the Department of Biotechnology and Nanomedicine at SINTEF Industry. And as for the rest of SINTEF, we're working in very close collaboration with both national and international industry and academia. And we work both in smaller direct contract research with industry and also in large international network projects. And basically how we work is like this. So the main goal for SINTEF is technology for a better society. And we want to solve challenges for industry. And we do this by, so these challenges that are found out there are driving the competence building at SINTEF. So we build competence, methods, and technology that is established through this research-led collaboration. And we, of course, use this expertise that we gain, again, to solve new challenges. So this is an ongoing process in SINTEF and basically how it works. Within our department, biotechnology and nanomedicine, we are working in also a very wide range of research fields. So from production and development of pharmaceuticals to aquaculture, food, and drug delivery. And we're very proud of our infrastructure platforms. We have equipment to grow microorganisms from milliliter scale up to 300-liter fermenters. We have a very modern molecular biology lab, which includes also sequencing facilities. We have numerous high-throughput robotic systems for cells, also including a confocal microscope and cell sorter. We have what I would say one of Europe's most sophisticated and advanced mass spectrometry infrastructure. And we also do more like kind of a hardcore chemistry, where we produce and develop particles, membranes, and do surface modification. But we're going to talk a little bit more about what we do with microbiota at SINTEF. So what we realize is that we have both competence and infrastructure that is highly relevant for a very wide range of the technologies that are needed to fill important gaps within gut microbiota research. And this is why we now are in the process of establishing a quite comprehensive toolbox within different selected areas of gut microbiota research. And this is to support both national and international research activities within this field. And the three main focus areas at SINTEF within microbiota are the following. As you see in the picture here, it's characterization and data handling tools. It's biological models, so both in vitro and in vivo. And finally, smart delivery systems. And we have just started now a strategic project in SINTEF, which is internally funded, where the aim is to build more competence, develop novel methods, and also adapt our current infrastructure to gut microbiota research. And this is to meet these needs that we see within the three focus areas here. And now we want to today present the activities in these different focus areas. And Tonya will start by taking you through the characterization and data handling tools. So go ahead, Tonya.
Yeah, and thank you. As Ihr already said, we have quite the comprehensive infrastructure. I listed some of the relevant equipment down at the side of the slide here, not really needing to go into the details. So we look at, for instance, the microbial potential of the microbiota, meaning which organisms are there, or which genes do they encode. So what kind of reactions can take place in the microbiota. We also look at microbial function, meaning which of those genes are actually active, which are turned on and off in different regions of the microbiota or in different samples, depending on health issues, for instance, patient samples versus healthy people. We can also look at the metabolites, the metabolome in the cells in the complete microbiota, which will describe the microbial activity. And if you also include patient information and other types of metadata telling you something about what type of samples are we investigating, it's possible to integrate and translate it into something we can interpret, meaning helping and aid diagnostics, for instance, or seeing how a patient will react to treatment. So during the implementation, we typically look at the microbiota in model systems, because if you're going to look how a compound reacts in the body, you'll have to first evaluate whether it's safe. So therefore, you can use in vitro samples and then later in vivo models. This you can use, for instance, to study pharmaceuticals, nutraceuticals, all these food additives or nutrients in general from food and feed. We can look at the biodistribution. We can look about the host microbial interactions and the relationship between what happens in the microbiota and the health level of the patient. Next. So if we go into the DNA and RNA sequencing activities that we have in our toolbox, one typical example is looking at what type of organisms are in the microbiota. For this, we use what is called metabarcode sequencing, in where you do amplicon sequencing of a short region of a target genes, and then you use that to evaluate which organisms are really there. And then, of course, you will receive a lot of patterns that you can look at, and then you can perform a clustering analysis where it's possible to compare samples, see if samples taken from one part of the gut or from patients differ much from other samples that you find. And that can be something you add to evaluate. You can also look at total sequencing of the DNA sample, which we'll see in the middle here. In that case, you look for what genes are encoded in the sample. The data I'm showing here are from a moose, or actually it's from 5 animals that were collected during the hunting seasons. So we only study the leftovers from the meat hunt. And here we see just how we find coding sequences, meaning the genes in the genomes, and then figure out which kind of enzymes or other proteins are encoded by these organisms, which can tell you something about the potential of degrading nutrients, for instance, the grass and other things that the moose has been eating in this case. And then you also can do RNA sequencing, which will tell you something about which of these genes that are encoded in the samples are active. I'm including data here from a culture study, which is from the production of fatty acids from a microorganism. But the same principles also applies when you're looking at the total gut microbiota. And here you can look for differences in activities between a healthy person, a sick person, before or after treatment, etc., to see how the microbiota responds. Next. If you go into the metabolomics part of the MS analysis, you can say something of the microbial activity in the cells. You can also look at the patterns to evaluate which kind of organisms are it specifically. Sometimes if you isolate individual strains, this type of method will give you a better resolution, telling you a lot more about which precise organism is it, what kind of strain is it, compared to other previously categorized strains. For instance, in epidemics. So if you have an outbreak of something affecting the gut and there is a spreading of pathogens, this can be used to figure out if the pathogen is in the sample. Next. Another interesting analysis you can do with MS is Maldi Imaging, where you can detect different nutrients or other compounds, chemical compounds in general, within different section of a tissue, or for instance, a colon or other material that you isolate. And you can then go through the entire area, scan it, and figure out, okay, where in this sample do you find, for instance, a drug or something else that you have added to the system. So people tend to like this one. And then, of course, you can look at the entire metabolites that you find in the sample, compare if there's a pattern difference between healthy versus sick or before after treatment, etc., to see how also drugs and other compounds are breaking down or affecting the cells. And next. Also, these types of analysis are relevant in general, so you can use them to look for toxins, for instance, or heavy metals that you can find either in the environment or in the food, or even in patient samples if they've eaten something that they're not supposed to eat or been affected in other ways, so it's affecting your body. Pesticides, toxins in general, or more positive things like fatty acids, vitamins, and other things that you want to find in your diet. So it's quite comprehensive, and you can find a lot about the both the status of the cells and what you eat just by analyzing this way. Next. And then we're going to move over to our cultivation and biological models. So as it already showed you, we do have comprehensive equipment for the cultivation of cells. It's ranging from the really small volumes. In this case, we have the biolector, which can run full cultivation studies in 1.5 milliliter scale. And it goes all the way up to the industrial range pilot scales, where we can, for instance, cultivate cells for the production of fatty acid, which is shown here as an example. And here that is for the production of omega-3 fatty acid for the fish farming industry. We can also use it to break down organisms or seaweeds and other things we harvest from nature to produce pharmaceuticals, or we can use it to cultivate the gut samples, like we can do in the biolector. In that case, you would run it under anaerobic conditions, so that the organisms in the gut that normally do not see too much oxygen will also thrive and be able to replicate. Next. Yeah, and we also have quite comprehensive in vitro models available. We work with the mammalian cell lines, so we can use them to evaluate nutraceuticals, pharmaceuticals, and other compounds for toxicity, for instance, or how efficiently they are taken up through, for instance, 2D and 3D cell assays. And some of these assays are also fully automated in the high throughput robotic laboratory. So you can run a lot of samples in parallel. Our labs are biosafety too, so we can also work with the multi-resistance microbial strains and also pathogens to evaluate how we, for instance, get rid of them. And as I mentioned, we also are able to run anaerobic cultivations, which is really relevant in this case when we're working with the gut microbiota. And within this new project that we are starting, we also will be acquiring and implementing a lab on a chip or gut on a chip version, which is called organoplate from Mimetas. And in this case, we can also study in more details how drugs are being transported from, for instance, a blood flow into the gut or vice versa, and how the pharmaceuticals that we do add, nutraceuticals, can affect the microbiota and vice versa. Next.
Yes, so in addition to the in vitro models that Tony was talking about, we also have quite comprehensive range of different techniques for in vivo studies, so in animals. And we have dedicated personnel that are highly experienced in this field, which is quite important. So we do all kinds of studies from treatment and effect of pharmaceuticals and nutraceuticals, where we look at the efficacy, pharmacokinetics, and the biodistribution of pharmaceuticals, nutraceuticals, and metabolites of those. And this is done in various disease models of mice. It's also, of course, very important to look at safety of these substances, and these are studies that we also conduct, both short-term safety and long-term safety. We use different administration methods to get these substances in the body. It's both intravenous and local injections. We use different surgical techniques and also oral administration. To be able to evaluate the effects and safety and so on, we use various imaging systems. So we have a whole animal imaging system. We use ultrasound imaging. We also have animal PET MRI. As Tonje showed, we also have mass spectrometry imaging facilities and multiphoton and confocal microscopes also to look more on ex vivo tissue from mice. Of course, we can do microbiota analysis in these rodents to look at the direct effect of microbiota. And now we are implementing currently different novel models also that are relevant for gut microbiota, including inflammatory bowel disease and colorectal cancer models in mice. So finally, to the smart delivery systems. So what do we mean by smart delivery? Well, mainly we're talking then about encapsulation of substances in micro or nano capsules or particles. And these can be bioactives, small molecule drugs, or it can be larger substances or even live cells. And what we can achieve by such encapsulation is various things, ranging from lower toxicity. We can get a targeted delivery, more of a controlled release, so getting things to the place where we want them to have an effect. We can combine several compounds, increase bioavailability, and so on. So many advantages of doing such encapsulation. In SINTEF, we have been working with nano and microcapsules for over 35 years now. And we use mainly then degradable, both natural and synthetic materials, to tailor make nano and microparticles for smart delivery. We are mainly focusing on polymer and lipid-based particles and capsules and technology platforms that have been developed at SINTEF and in collaboration projects. We use these materials to make programmable release profiles so that we can adapt the release to the different applications and also, of course, do then release studies. We use various encapsulation techniques when we make these particles. So ranging from complex co-observation, spray drying, microfluidic devices that we have setups, emulsion systems, and so on. So it's a wide range of techniques that we use to make these various particles, depending on what kind of materials and applications we're talking about. We encapsulate all kinds of different things. So we have been involved in projects where we are encapsulating antibiotics, vaccines. We have very many projects where we're encapsulating anti-cancer drugs, but also anti-inflammatory and antifungal drugs. We have projects where we have been encapsulating human cells and bacterial cells in larger microparticles. And then in those cases, we're usually using natural polymers like alginate for encapsulation. We encapsulate nutraceuticals, vitamins. We have an ongoing project where we're encapsulating omega-3 and fish oils and so on. So the list is long. Of course, when we make these particles, we also need to do advanced characterization to find out. Can you hear me? I think the sound was, yeah. We do advanced characterization to look at the effects and the properties of these particles. So these are mainly then first physical chemical properties. We can use high resolution mass spectrometry to look at loading, payload, stability, release profiles, and so on. And we also have simple in vitro models for both gastric and intestinal release. And we then use various administration routes for these particles. So now we are adapting these methods for gut microbiota research. And this is then for targeting specifically the gut microbiota. And of course, the goal then is to promote human and animal health and welfare. And we have mainly for this 2 different approaches. The first is to do nano and micro formulations of bioactives. And the main goal is then to achieve both controlled release, so get the drug where we want it, or the bioactive where we want it, so to the right place at the right time. But also more drug the bug approaches where we want to target specific microbes in the gut and have a specific effect on specific microbes. So more of a targeted approach. The other approach is to encapsulate pre, and symbiotics, or immobilize them. And this also, of course, is highly relevant for gut microbiota research. And we're currently now developing particles and delivery systems adapted to this. So on a final note, as you have seen, we have both infrastructure and competence that we believe is highly relevant for gut microbiota research. And we're used to working in both small and large collaboration projects. And of course, we're open then for also novel collaborations within gut microbiota research, and hope that you will contact us if you're interested. And we look very much forward to hearing from Innovation Norway and the Research Council also when it comes to possibilities of funding and novel, exciting projects. So thank you very much.
Thank you, Ir and Tonya. It was really an interesting presentation. And what I can see from your project is that the SINTEF project is really a complete project. So it's really, really nice to have this in Norway. And I'm sure that a lot of attendees could be interesting to do some collaboration with you. So thank you. So now we are going to have our next presentation from Pauka Schatz. She is a PhD student at the Arctic University of Norway in Tromsø. Her PhD subject is the identification of anti-obesity and insulin-sensitive effect of Calmness oil, a novel wax ester-rich marine oil from Calmness firmicus. Sorry. She also has a background in marine biology and Arctic animal physiology. So now she's going to present us some of her last result from her PhD research. So please now, Pauk, you can start your presentation.
Thank you, and also thank you for asking me to present here today. Can you see my screen?
Yeah, it's perfect, thank you.
Yeah, so I'm going to present a paper that got published earlier this year that I worked on together with Kirsten Jansen, Jakub Mrazek, Alice Pedersen, Ragnar Olsen, and Terje Larsen. And the paper is entitled Obesity-Induced Alterations in the Gut Microbiome in Female Mice Fed a High-Fat Diet are Antagonized by Dietary Supplementation with a Novel Wax Ester-Rich Marine Oil. So that's quite a mouthful. So before I'm going to start presenting the results that we got in this paper, I thought I should maybe first give a brief introduction on what this wax ester-rich marine oil is. Yes, and that is called Calmness oil. And as Rachel just said, that is an oil extracted from the zooplankton Calmness firmicus. You see a picture of this little animal here on the left. And as I said before, it's a wax ester-rich marine oil, and that means that it's a bit different from other commercial oils that we have on the market today. So in this very simplified figure, you see the 4 main marine oils and their main or most important lipid class. So we have krill oil, and the most important lipid class in this oil are phospholipids, where the fatty acids are bound to a phosphate group. And then we have fish oil, in which the fatty acids are either present as or in as triglycerides. They are bound to a glycerol, or as an ethyl ester, where the fatty acids are bound to an ethyl group. But in Calmness oil, the fatty acids are bound to a fatty alcohol, and that's a lipid class called wax esters. Then, if we go a little bit further into the details of this Calmness oil, in this table here, we see the composition of the different lipid classes given in milligrams per gram lipid of the commercially available oil. And as you can see, the main, like more than 85% of the lipids are present as wax esters. And then there's a small amount of triglycerides, cholesterol, and free fatty acids. And I said that this is the commercially available oil. The composition is slightly different in the oil in the animal, and that's due to the enzymatic hydrolysis that takes place in order to extract the oil from the animal. And then in this little figure at the bottom, we see an example of a wax ester. So we see DHA, the omega-3 fatty acid, bound to the monosaturated long-chain fatty alcohol docosahexaenol. So this is the composition of the lipid classes. And then if we look at the composition of the fatty acids also and fatty alcohols, also given in milligrams per gram lipid. And then if we specifically look at the omega-3 fatty acids, so stearidonic acid, EPA, and DHA, we see that in particular of EPA and DHA, the concentration is relatively low if we would compare that to other commercial oils. For example, if you take one portion of Müller-Tran, that contains 600 milligrams of DHA and 400 milligrams of EPA. And in Calmness oil, this is only about 10% of that. But Calmness oil is very rich in stearidonic acid. It's one of the most richest oil, both terrestrial and marine oils, in stearidonic acid. And in addition to that, it's also abundant in several long-chain monosaturated fatty acids, such as this oleic acid, condoic acid, and cetoleic acid, which also have important health properties. And in addition, we have the fatty alcohols, and of particular importance are eicosanool and docosahexaenol. And it is thought that these fatty alcohols are transferred to their fatty acid, to their corresponding fatty acids, so the monosaturated fatty acid, condoic acid, and cetoleic acid, upon ingestion and then digestion and absorption in the body. So that's a little bit about Calmness oil. And as the title of the paper said, it is an anti-obesogenic. So I'm going to have a few slides on how Calmness oil can be used as a preventative or treatment in relation to obesity. This is a figure from a paper by Hupper et al. From 2013. This is, she did an animal study on male mice, 7-week-old male mice, that she fed 3 different types of diets. Either a control diet, which are these open circles that you see in the bottom line. And in this diet, 10% of the energy came from fat. She gave them a high-fat diet in which 46% of the energy came from fat, and those are the closed circles, the top line. And then we have a diet which is also a high-fat diet, but it's supplemented with Calmness oil. So in this diet, 1.5% of the fat, so of the lard, was replaced with 1.5% of Calmness oil. And this was either given, this type of diet was either given in the start, so from time point 0, and those are the up-facing triangles. And this is the preventative group, or they were given after 7 weeks of unsupplemented high-fat diet, and this is the treatment group. And then the time period was, as you can see, 28 weeks. As you can see, there's a significant increase in body weight when the mice were fed a high-fat diet compared to the control. But in both the groups that were fed this supplemented with Calmness oil, there's a significant decrease in the body weight development. And if we then look at these figures, on the most right, we see the body weight gain. And we see that this CAP, that's the Calmness oil preventative, and CAT is the Calmness oil's treatment. So where they were given this Calmness oil from time point 7 weeks. we see there's a significant decrease in the body weight gain in relation to the unsupplemented high-fat diet group. And then if we look at this middle figure, where we look at the PWAT stands for perirenal white adipose tissue, which is the fat depot in the body. So if we compare the fat depot in relation to the body weight, it's also significantly lower in the Calmness oil treated groups compared to the unsupplemented high-fat diet group. And also the adipocyte size, that's the size of the fat cells, was significantly lower. In a follow-up study, they looked in 2014, they looked specifically at not only at the Calmness oil, but specifically at the wax ester component of this oil. So they had another feeding experiment, also with 7-week-old male mice for 28 weeks, where the mice were either fed a unsupplemented high-fat diet, or they were fed a high-fat diet supplemented with 0.2% of purified EPA and DHA as an ethyl ester, or as 1% with wax esters derived from Calmness oil. And these, the diets were composed in such a way that the total amount of omega-3 fatty acids were the same in the ethyl ester group and the wax ester group. And as you can see, there was no difference between the in body weight development in the high-fat diet and the high-fat diet with the ethyl ester groups, but there was a significant decrease in the high-fat diet group that was supplemented with 1% Calmness oil-derived wax esters. So that shows that from the Calmness oil, it is mainly the wax esters that have this body weight reducing effect. And also then if we look not only at the body weight, but specifically at the fat, that's also lower, as you can see in the left figure, in the wax ester treated group. So that's a bit on the background of what Calmness oil is, and particularly in relation to obesity. Then to the results, or like the paper that I'm presenting, that we published earlier this year. So it is well known that alterations in the composition of the gut microbiome have been linked to several pathologies, including obesity-related metabolic disorders such as insulin resistance or diabetes or cardiovascular diseases. And since Calmness oil has this weight-lowering effect, we had two aims. First of all, we wanted to know if this diet-induced obesity has an impact on the composition of the gut microflora. And then second, we wanted to know if this Calmness oil can modify any obesity-induced alterations in the microbiome. And we wanted to compare this to a known anti-obesogenic, which is, we used exenatide, and that's a GLP-1 receptor agonist, and it's also used for the treatment of insulin resistance. And you can see a figure of that here below. And we were particularly interested in this because it is thought that the wax esters in the Calmness oil are hydrolyzed in a later part of your digestive tract, and that you're absorbing then the fatty acids also in a later part of your digestive tract, so in the colon. So we thought that because of that, it will also could change the microbiota. So what we did, we had a 7-week-old female mice. The other papers or other figures that I showed you were done on male mice. And these were female mice, and they were fed a high-fat diet for 12 weeks in order to induce obesity. And then in the following 8 weeks, they either received 2% Calmness oil, rather than the 1.5 that we saw in the figures before, or they were administered subcutaneous administration of exenatide with 10 micrograms per kilogram body weight per day. And then we also had a group of mice that was fed a normal chow diet or a non-supplemented high-fat diet as the lean and obese controls. And then at the end of this 20-week treatment period, we collected the feces from the colon, and the microbiota was analyzed using 16S sequencing. Then these are our weight gain results. The open circles are the normal control diet mice, and then the squares at the top are the high-fat diet fat mice, and then the open, or like the upward-facing triangles, that is the mice that were fed high-fat diet supplemented with Calmness oil. And as you can see, there's no difference between in weight gain in this group and the unsupplemented high-fat diet group, unlike what we had seen before. And that might be because either these are female mice and they respond differently than the male mice, or because the time period of this experiment was 20 weeks instead of 28 weeks. But although we didn't see any change in weight gain, we did observe a change in the fat, the adiposity index, so the amount of fat inside the animal, and that was significantly lower in the Calmness treated group. So because of that, we still call this an anti-obesogenic. And then the downwards facing triangles, the black ones, those are the mice that were given a high-fat diet and had exenatide treatment, and that was significantly at a significantly lower body weight gain, as expected. Yes, and then to the microbiota results. So these two figures show the profile of the microbiota both on phylum level and on genus level. And in the phylum, we see that the most abundant phyla are the Firmicutes and the Butyrate, and that is also what we expected. In most papers, these ones are the most abundant. And the profiles there are a slight difference maybe in the exenatide treated group, but other than that, they're relatively similar. But then if we zoom in more to the genus level, the lower graph, we see quite some differences. For example, we see it in the normal control diet group. There is no yellow bar, which is the Lactococcus, and Lactococcus is considered a pathogenic bacteria. Whereas in all the high-fat diet treated groups, there is a yellow bar. And we see that, for example, in the exenatide treated group, the profile is more similar to the normal control group than the Calmness treated group. And then we see, for example, in the Calmness treated group that this green bar, which is Lactobacillus, is bigger than in the other groups. And Lactobacillus is considered a health-promoting bacteria. So this is more on first glance if you look at the profiles. We also did some statistics that you see here in this bar graph. So the red arrows indicate that there is a significant difference in the abundance of these bacteria between the high-fat diet group and the normal control diet group. And the yellow bar arrows indicate there's a difference between the high-fat diet group and the high-fat diet supplemented with or treated with exenatide. So in the normal control diet group, we see that there's a significant increase in the abundance of Alobaculum, but a decrease in the abundance of Lactococcus and Leuconostoc compared to the high-fat diet group. And then we see that if we treat the animals with exenatide, there's a significant decrease in the abundance of Lactococcus and Streptococcus compared to the unsupplemented high-fat diet group. And we didn't see any significant changes between the high-fat diet, the unsupplemented high-fat diet, and the high-fat diet supplemented with Calmness oil group. But just because something is not significantly different doesn't mean that it's not biologically relevant. So because of that, we also had looked at the data in a different way. We generated this heat map. And what we see here is that each column indicates the feces from one particular mice. And then on each row, we see the different bacteria genus. And then the color of the squares indicate the relative abundance of that particular genus in that mouse compared to the abundance in the other 19 mouse. So on average of these 20. So the genera I just mentioned, so like for example, Alobaculum, we can see that, you can try to, if you can see my mouse. Alobaculum is, as was also present in the other figure, more abundant in a normal control group compared to any of the other groups. But so is, for example, Oscillospira, which is higher abundant in the normal control group compared to the others. But this was not significantly different. And then we saw that in the first graph that Lactobacillus was more abundant in the Calmness treated group compared to the others. And although this was not significantly different, we still see that in there's a higher abundance on the heat map compared to the other groups. And the same, for example, for Bilophila. This is also a pathogenic considered bacteria, which is almost not present in the Calmness treated group, but it is present in some of the other other diets. Yeah. Then some conclusions. So our first aim was to know if a diet-induced obesity has an impact on the composition of the microflora. And we found that a high-fat diet changes the microbiota composition in an unhealthy direction due to enrichment of the pro-inflammatory Lactococcus and depletion of the anti-inflammatory and health-promoting Alobaculum and Oscillosphera. And our second aim was to know if a dietary Calmness oil or infusion with exenatide modify any obesity-induced alteration in the microbiome. And we found that dietary Calmness oil was not able to restore the microbiota composition to that of the normal control group, but it did show a relatively high abundance of Lactobacillus, which is considered a health-promoting genus and often related to weight loss. And there was a decreased abundance of the pathogenic considered Bilophila. And that treatment with exenatide partly restored the bacterial profile found in the normal control diet group, and it seemed to reduce the abundance of Streptococcus and Lactococcus, which are both considered pathogenic. My face, yeah. Yeah. Oh, I'm quick. Yeah, and this is the last slide. So if you want to read more about it, you can find in this paper here, or just send me an email, or yeah, I guess in the question round at the end of the webinar. Thank you very much.
Thank you, Pauke, for this presentation. It was really, really nice. So what I understood is that we have to take some pills from of Calmness oil. That's right.
Yeah, maybe, but more the men than the women if you want to lose weight. But yeah.
Okay, and but I just have a question. The product, the Calmness oil, is available on the market already?
Yes. Yeah.
Okay, so we can find it in Norway or in EU?
At least in Norway and in some other countries in the EU, but maybe at least.
Okay, so that's good to know.
Yeah.
Before Christmas, take a small pills to lose some weight. We have a small question because we are a little bit early, so maybe you can answer. So we have a question from the attendees. Do these mice eat the same amounts of their feed to explain the differences in gain?
Yeah, they do. We weighted the food intake, and there were no differences in that.
Okay, and so it's because you can weight all the food intake for each mice?
No, not for each mice. We have them in 3 mice per cage, and then we weigh the amount of the food that we give to them, and then we weigh the amount that that they do not eat, and then we can see the difference. So then we know that that's what they ate per 3 months.
Okay, so thank you again for your presentation, and please you can ask some question to Pauke directly in the Q&A or directly in the chat, and we will have a small discussion after the presentation. So now we are going to have a presentation of funding, how we can have some funding from our partners. So Arthur Almestad from the Research Council of Norway and Marie Klock Leita from Innovation Norway are going to introduce us to potential funding to support R&D projects, but also to support projects between academicals and industrials. So please, Arthur, if you want to start to share your presentation.
Yes, do you hear me all?
Yeah, perfect. We hear you and we see your screen.
Fine. Yes, thank you for the invitation and introduction, Rachel. I have prepared a short presentation that focuses on the Research Council funding opportunities, both for R&D institutions, but most for the companies. But before I go further, so a few words about our organization, the Research Council. We are the key advisory body to the authorities on research policy in Norway. Our aim is to promote a society where research is created, used and shared, and we invest a wide range of research and innovation projects, from basic to research-based innovation and also commercialization from the universities. approximately, we invest about 11 billion Norwegian krone. This is 120 million euro in research and innovation annually. It's our task to ensure that the funding goes to the best research and innovation project. I think I will start with this figure. It shows that how much we, since 2016, have invested in the project where gut microbiota is mentioned in the summary of the project. Many of these projects are related to human health, but many are also related to fish farming and animal welfare and health. The source for this figure is our project bank, where you can find and read a summary of all the projects we have funded. You will find here 35,000 projects in total, and this is open for everyone. We have invested, as you see, 328 million Norwegian kroner to gut microbiota projects in the period from 2016. And mostly of these projects are run by R&D institutions. But we also find examples of projects where companies own the projects and where R&D institutions are partners. We have seven different application types in the Research Council. In the calls for proposal at our website, you will find all the relevant practical information of these types. We have 4 main application deadlines per year, as well as a number of funding schemes with open-ended deadlines. Applicants compete with another for funding. First, researcher project. Only approved Norwegian research organization can apply these application types. Total funding under a call with deadline in next February, is 3.4 billion Norwegian kroner. This call is for researchers who can demonstrate the ability to conduct research of very high scientific quality. And many of the projects on gut microbiota you will find in these types of projects. And then we have collaborative and knowledge building projects. These projects are to develop new knowledge and generate research competence needed to society or the business sector to address important societal challenges. Projects under these application types require collaboration between researcher groups and relevant actors from outside the research sector. And then we have, the total funding under this call with deadline in February is 1.3 billion Norwegian kroner, I think it was that. And the next is innovation projects. This project type is divided into 2, one for the industry and one for the public sector. The Research Council have now announced 1.3 Norwegian kroner billion in grants for innovation projects in industry for next year. The aim to this project type is to stimulate companies in industry to invest in and utilize R&D as a crucial part of their innovation strategy. This type is very relevant for companies in marine ingredients industry and where R&D institutions can be partners in the projects. I can tell you a little bit more this project. This is a project that is built on unique and innovative idea. And the result from the project may create value in form of new products, service, and production process. The companies collaborating in this typically have a need for new knowledge or new technology in order to deliver the intended innovation. And R&D suppliers may be research organization. In addition to this, we have also SkatteFUNN. I think most of you have heard about that. It's a right-based tax deduction scheme designed to stimulate research and development in Norwegian companies. And we also have industrial PhD funding scheme that is also very relevant for you. This is smaller research and development projects carried out as a PhD project and conducted by a company employee. This is also a project with a clear thematic relevance for the company's business activities and initiated and carried out in a line with the company's need. Further, we have also this commercialization project, and this is also open-ended. And the aim of this application type is to contribute to increasing commercial exploration of public-funded research in Norway. In short, we want to see more of spin-off from the research, from the R&D research from the universities and the colleges. Further, we have research centers. We're funding a lot of research centers. And example of these are a Center for Research-Based Innovation, and this is, as you heard, is based on innovation ideas and lots of innovation groups, researcher groups, working together with a group of companies. And we have SFF, that's Norwegian Center of Excellence, and this is about basic research. We are also funding research infrastructure for the research organizations, and at last we have also coordination and support activities, a lot of different types of that. So I think I stop there. And I have also prepared some attachments, and you can see this after we have received this from Rachel, I think. So I say thank you.
Yeah, thank you, Arthur. Yeah, the presentation will be available after on the website of Marine for Health. So if you want to have a look on this attachment, it will be available, I think, Monday. So thank you, Arthur. It's really nice to see that the Research Council of Norway already have a lot of funding project for the gut microbiota. That means it's an interesting topic for you.
Yeah, so take a look at this bank.
So now we are going to hear a little more about Innovation Norway from Marie Klock-Leite. So if you want to start, Marie.
Yes, do you see the right version now of the presentation?
Yeah, you can put it in a full screen if you want.
Yes, I have already done it, but that is on the wrong screen.
On the right, yeah. Oh, no, we don't see it.
Yeah, I know. I just need to do it one more time. I'll try one more time. I hope this will help.
Perfect.
Okay, perfect. Yes, hi, my name is Marie Klock-Leite. I work as a cluster advisor here in Innovation Norway in Møre og Romsdal. And now I'm going to give you a brief introduction to possibilities in Innovation Norway. And a difference between Forskningsrådet, the Research Council, and us is that we have instruments and for companies who are rather close to the market, are ready for develop new innovative projects that they're going to commercialize. And Innovation Norway strive to give local ideas global opportunities. And this we do by working towards our sub-goals to create more successful entrepreneurs have more companies that achieve growth, international growth, and more innovative business clusters. So that's our 3 focus areas. And when it comes to projects, we facilitate projects in these kind of areas, both for enterprise startups, innovation and development in existing companies, and international expansion. And Innovation Norway is a sparring partner who offer both expertise, networks, and capital. And we do that through office structure around the world, both here in Norway. We have offices around in each region. Here in Møre og Romsdal, we have 2 offices in Ålesund and Molde, so we're working with businesses all over the region. And also, we have offices in over 30 countries today, and we have offices in actually the most of the markets where Norwegian businesses see market potential. Yeah, and when it comes to capital for projects that you introduced me with, Rachel, you can see on Innovation Norway more like an investor who offers financing to companies with innovative business ideas. When you wish to create something that's not in the market today, and when the company and the idea have a great potential for growth and internationalization. And when the project has high risk, Innovation Norway can reduce that risk and correct market failure. So you can think of Innovation Norway like an investor who love high risk projects. And the aim for us is to achieve return on investment through growth and value creation in Norway. And for which kind of projects we are looking for, that is, we are prioritizing innovation projects that have a great prospects, high degree of innovation. It has to be something new and unique, and that they have a likelihood of success. And we don't have any specific instruments for this area, but we have general instruments which fit. When you have these kind of projects, they can fit into our portfolio. And lastly, I just wanted to add which people who are working in the marine sector here in Møre og Romsdal, and that is Odd Reidar Kvalsund, who is his head of department for marine, and Ingvild Kjærstad, who lots of companies in the Blue Legacy already know, have a high competency in this area and work with lots of companies, and myself included. So this information you can find on the slides that Rachel will send you later on, and please contact us if you have anything that we can answer.
Thank you, Mary, for this presentation of Innovation Norway. So if someone wants to get some funding, they can ask you.
Yeah.
Okay. So finally, we are really proud now to welcome our last speaker, the Dr. Marius Trosseid from the Oslo University Hospital. He is a consultant in infectious diseases and is leading a research group on clinical microbiology and microbiota medicine. The name is CLIMIC at Oslo University Hospital. The overall scientific focus is the role of the gut microbiota in chronic infection, as well as inflammatory, metabolic, and cardiovascular diseases. Mr. Troseid has also established a regional multidisciplinary research network, REMIX, which is hosting a yearly national microbiota conference. So now, Mr. Troseid, you can share your screen with us.
Thank you. Can you hear me?
Yeah, we can hear you perfectly.
Thanks for the invitation, Rachel. Really nice that you're setting up this meeting, and I'm very happy to be invited. My name is Marius. I'm a medical doctor, a specialist in infectious diseases. So as you say, we are trying to establish the microbiota as part of the clinical practice. That's really our goal. So I'm heading this regional research network for microbiota science, which has a clinical focus. My closest collaborator, Johannes Hov, he's also leading a sister group, and together we're also trying to establish this as treatment at the hospital and other hospitals in Norway. So we're now working to establish a donor bank for fecal microbiota transplantation that should be certified in all ways. We are starting to use it in Clostridium difficile infection, but we'll also probably try to make it possible to for example, boost the effect of immunotherapy in cancer, and so on. So there's a lot of collaboration with different research groups with clinical focus. So we are now collaborating with rheumatologists, oncologists, cardiologists, infectious disease specialists, gastroenterologists, and so on. And I started out with HIV infection, which was my interest, and after realizing that it was very difficult to perform these studies and to get things analyzed, we have actually set up our own in-house microbiota platform. So we can run clinical trials, we can analyze the microbiome, and we also have 3 bioinformaticians now that can help us look into these results. I have to say that these days I'm heavily involved in COVID research. You know, that's the agenda, just so you know it. So I'm a part of a huge EU response now to try to make a big multinational trial running across Europe testing COVID drugs. But still, I think it was a very nice webinar here. So I think I really like to share our thoughts and hear what you think about it. So I think you've already been introduced to the microbiome, so I'll be brief here. Is it relevant for cardiovascular disease? I mean, as you work with marine products, this could be interesting. And finally, can we target the gut microbiome in personalized medicine and nutrition? It's very variable from individual to individual, and I will end my talk with discussing if a banana is always a banana. So I think you heard already that we are talking about a lot of microbes and a lot of genes. So we probably have the same number of bacteria as we have human cells in our body. And they have a much larger genome in total. So we're talking about perhaps 500 times the number of human genes. And it's also a very powerful organ. So it can transmit health and disease. And this study by Tmao, you've probably heard about it, or many of you heard about it. It really opened up the field now 14 years ago when he showed that if you transferred the microbiome from obese to slim mice, the lean mice became obese. The diet didn't change, but the microbiome changed to resemble that of the obese mice. So this is really contagious obesity, which was really a new concept in those days. And I think after that, we've seen that anxiety, cancer, a lot of other disease states can be transferred by the microbiome in susceptible animal models. And we heard from the Research Council that they are providing money for this. So I also want to thank the NRC for a lot of funding that our groups have received. You see that hits on microbiome in PubMed is really exploding. And part of this is really that this is a technology-driven research frontier. So when we tried with our first HIV cohorts to analyze it back in 2006, it was difficult because the methods were not really developed for analyzing big data. But after the Illumina platform came, this has really been much more feasible, and it's not very complicated to do this analysis now. But to interpret this, the data is more complex because we are so different on the inside. You know, genetically we are at least more than 99% equal, but the microbiome is really a fingerprint or a stool print, what do you want to say. So here we have sorted 117 healthy Norwegians according to increasing abundance of Bacteroides, which is the most abundant bacterium in our normal flora. And you see this varies from more than 50% to 3 or 4% in some adults. And these are healthy individuals. So we're really very different on the inside. So if we want to target this organ, we probably need to personalize. And also, I want to also point another thing. In most of the literature that has been published until now, we got the Illumina platform, which is analyzing the 16S RNA gene. So this is only one gene per microbe. And you use it to predict the abundance of microbes and the function, but it's really a very superficial tool. So if you want to go on and really see what's happening, you need to do metagenomic sequencing. But this is far larger data. But now we have submitted our largest clinical cohorts for metagenomic sequencing, and we'll find out the bioinformatics there. But it's more complicated and more heavy projects to run. And also connected to this, we also like to do metabolomic analysis to be able to predict what the microbes can do and then actually measure what they are doing and then connect this to clinical outputs. So I think this is the way to do the microbiome research from now. You need to do metagenomics, you need to have other omics, and you have need to have large clinical trials to do this. So it's really very demanding research, but I think you need to go that way if you want to prove anything. So I was asked by eBiomedicine to write a review on diet, the microbiome, and cardiovascular risk, which was published earlier this year. So I'll just share some this cartoon with you to point to some mechanisms that we think might be at play. So it's really an interaction between westernized food, the microbiome, and metabolites and toxins. That's really how we see it. And this is probably scratching on the surface, but this is what's mostly published in literature. So one thing is that a lot of disease states we see and risk states, we see that the gut is kind of leaky. So parts of the gut content can be translocated into the circulation. My daughter is homeschooling and I couldn't put up the background, so just excuse me. So in some of the disease states, we can actually measure toxins that are translocated, which can trigger inflammation and which can trigger a lot of inflammatory diseases, such as cardiovascular disease. And we know that LPS can translocate in the gut of heart failure patients or HIV patients, diabetes patients. But even if you guys go to McDonald's, you will measure, at least if you go for a high-fat, high-carb diet, it's possible to measure endotoxins in your blood up to 3 hours after this meal. I always take my kids to McDonald's when we drive to Sweden to visit my wife's family, and they love it. But I try to do this not every day, you know. I think if you have some bumps up and down in toxins, it's probably okay. But if you do this every day, what does it do to you on the inside? That's an interesting question. The control group here, they got an American Heart Associated certified meal, so they ate salad, nuts, olive oil, these kinds of things. And we know from animal studies that chronic LPS infusion can cause both obesity and diabetes. In this study, which is now 7 years back, we went to a cohort performing bariatric surgery. So we got adipose tissue biopsies during surgery from different compartments in the body. And we also had CT scans. We could measure the content of the different compartments. And we also measured the LPS and other things in the blood. And we really found a very close correlation between endotoxins in the blood and the amount of intra-abdominal fat, but not the other fat compartments. So this was really related to the intra-abdominal fat. We were also able to detect bacterial DNA in the biopsies, but only those that were in proximity to the gut. So I think we really showed here that the microbes are able to go into the adipose tissue, and the amount of adipose tissue is related to the level of toxins in your blood. So it's interesting. The metabolite that has received most attention in the literature is Tmao. It's really proposed to be a link between red meat and the microbiome and cardiovascular disease. For your interest, there are also certain kinds of fish that produce Tmao. So I think you find it in deep water fish. It has like an antifreeze effect. So if you're going to do studies on Tmao levels in your products, you might get surprised. I haven't read all the literature, but the story in the papers is that it's a link to red meat, but it's more complicated than that. So at least in the early studies, it was proposed that you had to have 3 steps to produce Tmao. First, you need to eat nutrients that contains either phosphatidylcholine or carnitine. Carnitine is from red meat, but vegans do not produce this metabolite. Second, you have to have an intact gut microbiome. So if you give 1 week of broad spectrum antibiotics, the production of this marker disappears. You need an oxidation step in the liver from Tma to Tmao. So you find in liver failure patients, they do not produce it in the same way, actually. And then it has been shown in animal models that precursors of Tmao, they are sufficient to trigger atherosclerosis, but not if you add antibiotics to the drinking water. So it's really at least an indirect sign that this is caused by the microbiome. And these are the 2 first papers that were published in the field. First from Stanley Hazen group, which has really opened this field. He showed that high levels of Tmao predicted myocardial infarction and stroke. We showed the year after that high levels of Tmao predicts progression in heart failure patients. So here you see in the upper tertile, 50% of our patients have progressed to either cardiac transplant or death within 5 or 6 years. So it's a powerful predicting tool. And the Stanley Hazen group, they really publish a lot of nice things. They also measured here Tmao in the emergency room. And they find that if you have elevated Tmao levels, it predicts whether or not you get a new myocardial event within 30 days, irrespective of troponin levels. Troponin is the cardiac enzymes that we measure in the clinic. So, so I wrote this editorial discussing if the microbiota is ready for the emergency room. We think this marker might react with platelets and give platelet reactivity and blood clot formation, but we're not there yet. It's still not a clinical tool, but I think the Stanley Hazen group really brings it closer to clinic. And the last marker I will talk about is butyrate. It's one of the short chain fatty acids. It's oxidation product from dietary fibers. And we see that if you have low butyrate levels, you tend to have more gut permeability. Sorry about that. So butyrate is really the main food product for the colonocytes. So it's very important for the integrity of the gut barrier. It also has immunomodulating effects. So it stimulates regulatory T cells in the gut mucosa. So it's an interesting marker. And the reason why we got so interested in this is we performed 2 years ago the largest microbiota study in heart failure patients from 2 independent cohorts. And we only reported those bugs that were differentially abundant in both cohorts after a correction for multiple analysis. And when doing so restricted analysis, we really found that, just minimize you guys like this. we really found that almost all of the microbes that were reduced in heart failure, they belong to the same bacterial families, Lactospiraceae and Ruminococcaceae. And these are really known to be butyrate-producing microbes. So this was clearly the strongest signal in this cohort. And afterwards, there have been several smaller studies finding a lot of the same traits. We also found that the total capacity for producing butyrate, the genetic potential, was reduced in heart failure patients. And interesting, we also found this to be potentially related to prognosis. So one of these famous group, Hali, was lowest in those that progressed to transplantation or death. In a later study, we have looked at food frequency questionnaires in this cohort, and we find that a lot of the dysbiosis that you find in heart failure, like low diversity, Tmao and observed OTUs, that's diversity measures, and a lot of these butyrate-producing microbes, they're actually correlated with dietary fiber intake. Most of these studies do not account for diet. So at least after this, we have added food frequency questionnaires to all of our clinical studies. But it could also, of course, be a clinical target. I mean, there are very few studies on fiber diet in heart failure or cardiovascular disease that are properly performed. We also found that Tmao correlated with intake of red meat, but not with the dysbiosis, not with the microbiota alterations we found in heart failure. So and looking at the literature, there's not a lot of Tmao being correlated with dysbiosis. So we think that Tmao is more a diet-dependent biomarker than a microbiota biomarker. So finally, can we target the gut microbiome in personalized medicine and nutrition? And the question is really, is a banana always a banana? So this very nice review, I don't think you can see everything, but it's strategies to target the microbiome. And there are several ways you can go. The most drastic is, of course, stool transplantation. That's what we are setting up now in the transplantation unit at the hospital. So we regard this as an organ, or at least a cellular organ. So the question is, how is it regulated? Is it a medicine? Is it waste? Or is it a transplantation product? So I think there is competition here. I think drug companies want this to be a drug. And I think for the benefit of patients and the health systems, it should be a transplantation product so that we can recruit donors and we can screen them like we do when we give blood products, and we could give the stool if we think that's right from a medical perspective. This trial, the fat loss trial, really it's a very old trial, but it's interesting. It's fecal transplantation to lose metabolic syndrome. So here they gave microbiota from lean donors at the left. This shows the insulin sensitivity. So these are lean controls. And then the gray ones are those with metabolic syndrome. So these guys here, you see they have decreased insulin sensitivity. They give stool from the lean ones to these ones, and you see that the insulin sensitivity is normalized. In longer follow-ups of this was reversed. Normally the microbiome will go back to your basic afterwards. But for example, in Clostridium difficile, where one bad bug has taken control, 1 or 2 transplants is sufficient to cure the disease. But if you want to cure a chronic disease, you will probably have to repeat the fecal transplants or do something else, or maybe combine with some dietary interventions. So the other end of the scale, the most elegant one is, of course, not to replace all the bugs, but to drug the bug. You don't drug the patient, but you drug the bug. So here, this is a targeted molecular inhibition of microbial trimethylamiine synthesis. So this Tmao story that you heard about. So it's possible to target the enzymatic steps in the bugs that produce Tma before oxidation to Tmao. So by targeting the microbes with a Tmao inhibitor, the Tmao levels were reduced and atherosclerosis was reversed in these animals. So this didn't kill the microbes, it just drugged the bug without killing it. So it's a non-lethal intervention. I haven't seen it proceed to clinical trials yet, so there might be some difficulties here. And for now, it's maybe easier to change your diet, I think, if you want to target this. So this is a trial that measured Tmao levels in healthy individuals. So first, there's a huge variation at baseline, and then they do a non-meat intervention. So all are asked to eat vegan foods, and you see Tmao goes down, and then they have a red meat challenge, and you see Tmao go up. And then the most interesting thing, I think, is that then they switched from red meat to white meat, and the Tmao levels went down. So this is really a red meat biomarker. Another way of doing it could maybe to use a high fiber diet to increase the butyrate levels. As I told you, the butyrate is a short chain fatty acid. It's an end product of dietary fibers. So there have been studies showing that soluble fibers increase butyrate producing microbes, and also that it can lead to improved glycemic controls in type 2 diabetes patients. And finally, I need to show you this very nice publication in Cell. Is 5 years ago now, but still it stands as a very strong paper. Personalized nutrition by prediction of glycemic responses. So here they have looked at how can you shape the nutrition so you avoid too much increase in your blood glucose in order to reduce the risk of diabetes. Because, you know, sharp fluctuations in blood glucose are considered unhealthy in this way. So, and the question here is, what is healthy for you? So first, everybody registered their glucose levels with this subcutaneous sensor, and they registered all their meals real-time by a smartphone. So it's really a very large data capture here. And then they have measured the fluctuations in the glucose, and also they have looked at all the meals that have been eaten by all these participants. And first, the conclusion is that a banana is not always a banana. There was actually an extreme individual blood glucose difference after intake of different nutrients. So look here at participants 4, 4, 5. There's a rapid increase in blood glucose after eating a banana. Then it goes back to normal. But there is no signal after eating a cookie. Then have a look at 6, 4, 4. He increases after eating a cookie, but no signal after eating a banana. He's banana resistant when it comes to glucose. So I think dietists that talk about, glycemic response to different food items, I think they need to take into account that this probably varies from individual to individual. And then they wondered, could this partly be caused by a variable gut microbiota? So they entered here in a machine learning way, very groundbreaking research, I think. Microbiota, metagenomic data, blood samples, nutritional data, and anthropometry. I mean, weight, waist, hip ratio, and so on. And then they had a training cohort to try to predict increase by each individual, looking into the Firmicus, for example, the BMI, HbA1c, and so on, in these boosted decision trees. And then they had a validation cohort to validate if this really worked. And what they found was that the prediction was much better after using these machine learning strategies. And finally, they had this very interesting man versus machine competition. So here they invited dietists to give nutritional advice to some of these individuals. And they competed then against this predictor-based nutritional advice. So these got individual advice. So here some were recommended potatoes and others pizza to stabilize their blood glucose. But the the machine didn't outperform the dietist, but it performed just as well as the dietist. So to conclude, the gut microbiome is definitely altered in cardiovascular disease. Inflammatory and metabolic pathways could be targets for interventions, but I still think we're scratching on the surface. So we need to learn more through these multi-omic analysis and also to do good interventions, I think. The individual variation is substantial. So if the gut microbiome should be clinically relevant, a personalized approach is probably necessary. So this really points to personalized medicine, which is really a strategic vision, I think, from those who shape the health services. So thank you.
Thank you, Marius, for this presentation. It was a really, really nice presentation, really interesting. A lot of information. And I think as you conclude, as you say at the end of the presentation, it's that the microbiota, we're just scratching, and it's hard to have all the information, and a lot of companies are interested to study more on the microbiota, but we don't have access to all the data. And there is still a lot of challenges to have this information. And actually, I have a question for, I think, all of you. I think it's hard to study the human microbiota because you can have access only to the feces. So how we can have a global image of the microbiota?
I think if you want to really know what's going on, like for example in HIV, which is the disease I'm most interested in, you need to have biopsies, both from the end of the small intestine and the large intestine. So we have performed one study like that at Ullevål Hospital, taking biopsies before and after probiotic intervention. But it's of course very demanding for the patients, but I think that's the gold standard. Then you see what's in the lumen, you see what microbes are attached to the mucosa, and then you can also assess what happens in the mucosa, and you can do epigenetic analysis and so on. But it's of course very demanding. So as a clinical tool, that will not be feasible. But I think to understand the field, I think you need to go that deep actually, and to get obviously the small intestine, it has a very different microbiome. It's more related to the oral microbiome, but there it's very difficult to get patient samples. So I think it's interesting. It's what we want, but it's not feasible. Mm-hmm. No, I think this is something that the community needs to work on. I mean, you can have stool samples. You can maybe also have rectal swabs where you can get some of the microbes that are more attaching to the mucosa. We have looked at one study like that now for see if you can diagnose precancerous lesions for anal cancer, but it's the microbiota content is smaller on these samples, so it's harder to go deep in the sequencing. But I think what you need to do is really first to define what is disease-related microbiome, and then maybe first go very deep and then narrow down. So I think that's the way to do it if you want to take it clinically. You need to first understand on a metagenomic, very deep level, what are the relevant microbes, and then maybe you need to have a targeted analysis of these microbes with rapid tests. I think that's how it might look. And I also think that you probably need to look into this in different compartments of the body. So maybe the stool, maybe the spit, maybe the blood, maybe the urine. But of course, clinically, you cannot always sample everything from all the patients. But I think you need to see the crosstalk across anatomical compartments and then try to see if you can find some useful biomarkers that you can use clinically.
But the faces give like a first information, and after, if you want to go deeper, you can look at more specific organ or specific targets.
Yeah, I think that would be the way to go. Yeah, okay. And maybe also to find specific metabolites that you think are carrying the risk. And I mean, if you could pave the way in the gut and then identify some circulating markers, it would be easier to perform it in the clinics. I think one of my PhD students, she is now looking into extra cellular vesicles and see if they can carry some of the bacterial proteins and also other signals from the gut. But it's a hard work to do, so it's I think we are far from being able to use it.
Okay. Yeah, I have just a small question. It's a curiosity question, I will say, because what when you present the result of the fecal transplantation, it's really interesting, and the results are really amazing. And as you say that you have to repeat the transplantation for chronic diseases like for diabetes or for cardiovascular diseases. How many times? Like it should be like once a month or once a year?
These studies have never been performed, but what we see in different disease categories is that if you do a FMT, a transplantation once, normally it goes back. So there was also a group in California who tried this in HIV patients, and they see that the patients engraft, so they get a new microbiome, but then when they repeat the samples after a few weeks, it's back to basic. I think the one group that is working with it now very clinically is the rheumatologists, is Anna Hoffmann-Woll at Oslo University Hospital. She's looking at this systemic sclerosis disease, that Gunhild Stordalen is one exponent for. It's a disease where you get sclerosis in your inner organs, including the gut. So they have a lot of gut symptoms. And Anna and her co-workers, they performed a pilot study that showed that I think it was 1 or 2 transplants really reversed a lot of symptoms. So it could reverse incontinence in a lot of these patients. So I think it's worth trying out, but probably you will have to repeat it. And I think if you have incontinence because of a dysbiosis, you will be more motivated to get repeat transplants. Okay. If it's to prevent obesity, I don't think that's the way to go.
So we have a question from one attendee, actually a French also, from Pierre-Yves Morvan for PAOCA. So the question is, the bacterial genus Lactococcus and Lactobacillus decrease in obese mice vs normal, since Allobaculum and Oscillospira increase. Is it the same in humans? So from Pierre-Yves Morvan from Codif.
Well, we would have expected that there was like an increase in the lactobacillus in the normal control compared to the high-fat diet group. But that's based on like other literature I've read on mice. But I think in humans it's very different. Like it can differ from study to study. Like some papers say, so okay, we see this is a health promoting, and another one is other papers say like, oh, we saw, or maybe it's not health promoting, but we saw a decrease in the control group or something. So it's very different, but I think in general it would be the same, if you can say that there is a general, because that's there's not like a standard. It's difficult to say there's a standard. It also depends a bit. Like the results would be different if you analyze your samples different, or if you use a different reference database, for example, when you're analyzing your samples. So I think in general, I would say yes, they would be the same, but of course there are differences depending on the study that you're doing or the paper that you're reading.
Okay, thank you. Maybe some of you have questions for each other. I know that Marius, you will not be there at the beginning of the presentation. So Paik presents us some effect from Calmness oil on the gut of mice, and Tonya and Ilse present us the new project from Syntef.
I know Iri from Sintef, so we have started to have a strategic collaboration. So I'm glad that you're here as well. You're muted. You're still on mute, Iri.
Yeah, you're mute here.
I was trying to push the button, didn't work. Yes, I'm very glad that we have started this collaboration. I think it's a start of something bigger as well. So, and I was also saying to the rest that that we are in the beginning of building this competence in Sintef and looking for nice collaborations that we have just started with you guys, and you have a very large interesting network as well. I think there's a lot going on in Norway, and there's a lot of potential in increasing that activity as well in Norway. So I'm looking forward to that.
I think what is maybe a bit unique in Norway is that we have this tverrfaglig. I mean, this cross this network across specialties, and so I know a lot of Swedish and Danish scientists have also asked if they could join some of the meetings, and we always invite speakers from abroad, and I think they do not have the same kinds of network in most other countries. So that's nice thing about being in a small country. I think that yes, you can build this network across specialties, and then maybe you can also get some new research angles and maybe some unexpected angles. So I think it's interesting to have these kinds of interactions.
Absolutely, I agree.
Thank you. Actually, I have a small question for Pauka. My question is about, we can see that there is a decrease of the body weight gain in the obese mice after the supplementation with the krill oil. But my question is, the change of the microbiota, do you think is due to the krill oil directly, or it can be also due to this body weight decrease?
Well, we didn't really see a body weight change like in our study, because the weight of the animals was almost the same. It was just that we saw a difference in the adiposity index, so in the amount of fat that the mice had. But that is a very good question, and like I don't exactly recall it now, but it's still difficult to say like what comes first, like if you change your microbiota because you change your body weight, or you change your body weight because the microbiota has changed. But there have been definitely some studies done on that, and yeah. But in our case, there was no significant difference in the body weight between the Calmness supplemented group and the unsupplemented group.
And this is a general question, I think, how we can be sure that the benefit effect that we see is due to like bioactive compounds, or is due to like more a modification of the body, or?
Yeah, I mean, there are some studies done where they when they give antibiotics, so then you like, of course, you change, like you deplete the microbiota, and then you see differences in the body weight development afterwards. So that definitely indicates that there is an effect of the microbiota on and then changing that the body weight. Yeah.
And maybe here, with your expertise on the target molecule, it is possible to target specific cells and to see.
Yeah, it's that's also a good question. It depends a lot on on the cells, because we are working a lot with targeted approaches, for example, towards cancer. And targeting cancer cells is difficult because they resemble normal cells a lot. So so that is more difficult. Targeting bacteria is easier because they are very different from human cells. But then, of course, if you want to target specific bacteria, which would be the aim here, that is also, of course, a challenge. So yeah, but that's something that I mean, within the nanomedicine field that I'm working in, there's a lot going on such targeted approaches. So many different technologies exist and sophisticated technologies to be able to have this targeted approach. So that's something we're looking into.
Yeah. And Marius, maybe you can tell us which kind of bacteria we should target.
No, that's impossible to say. That's impossible to say. And no, there are so many of them, and I think I think normally you say that you can see that if you remove one bug from an ecosystem, it will be filled by some others, and sometimes if they have the same function, it maybe doesn't matter. If you wipe out the functional capacity of a part of the microbiome, it's maybe more important. So I think rather than focusing on each bug and what the name is, I think you need to focus on the function of these bugs. And you know, in humans, they do not exist alone. They exist as part of huge ecosystems. So and they talk with each other. So if you try to hit one, I think you will tell the other guys to speed up. So it's I think it's a bit more complicated. But I think also if you do studies on weight loss, it could also be interesting to look at calorimetry measures of the fecal samples. And you could also use some statistics, I think, to try to predict what causes what. But of course, it's always a bit difficult. But at least you can do regression analysis. Mm-hmm. We only did simple correlations, you know, in our food frequency questionnaire paper. But it was very obvious that a lot of the microbes that we singled out as being bad for the cardiac health, they were related to what you ate, actually. So. And then you can ask, do you have a dysbiosis because you have a cardiac disease or because you eat unhealthy? So, and I think to really look at the direction of cause effect, you need to have good models, so you can stimulate either in vitro, ex vivo, or in animal models. But if you want to take it to the clinic, you need to do randomized trials. Randomized controlled trials. So I think that's the only way of proving it in the end. If you can't prove anything in a randomized controlled trial, it will never be treatment. So then you can of course have food items that tastes good and are probably healthy. But if you want to go into the medical field, you will need to prove things with the same amount of evidence as for a drug.
Thanks. I have maybe another big question for you. Maybe it's too soon for if someone can answer, I don't know. What do you think about the use of marine ingredient of seafood? Do you know if there is some result from seafood effect on the microbiota? And what do you think about their potential effect on the microbiota? I know it's a hard question.
I'm not sure. We had an intervention study at Ullevål, or they had it. I think I referred to Professor Helseeffekter, who is doing these kind of studies. They had a prevention study, the diet omega-3 and intervention trial that showed at least borderline effect of omega-3 fatty acid intervention on mortality in an old study. They didn't collect microbiota data there, but I think there are, at least they will look at markers of microbiome and leaky gut in that trial. So that's a huge randomized trial. So that could give some information. But I also think that the same group now has published, at least I read in the newspaper, the OMEMI trial. It's also omega-3 intervention trial in elderly patients, and I don't think they found a lot of results there. But I'm infectious disease specialist, so this is really not my field to be authoritative about. But that's what I know about Norwegian research. And for krill oil, for example, I think some of these products might, as I said, Tmao is not only from red meat, it's also from some deep seafood fish, and I think also a lot of these marine products like krill, they might also produce quite high levels of Tmao. So I haven't read all these papers, but I think it's an interesting research field, but there's probably a lot to be done. So, yeah.
Thank you. And Baukak, what do you think about marine ingredients and microbiota? Because there is more research about omega-3 and lipid from marine ingredients, but what do you think about the protein side?
The protein side. I don't know that much about that, but I do think that there's a lot of the studies that you read looking at microbiota and marine oils, especially looking at omega-3 fatty acids, whereas like that's not the only thing that you will find in the oil. So it might also be interesting to not only look at that, but just look at the oil in general, and maybe look at the specific fatty acids that are present in. If you have like the commercial oil where you have like only the isolated EPA and DHA, that of course is interesting. But if you have like a cocktail of fatty acids, then that have probably a different effect on the microbiota. So that's something that I think is very interesting as well.
Okay.
I don't know so much about the proteins. I'm sorry.
For me, actually, I work more on proteins than on lipids. So, but it's always difficult to target one compound and to not look to all the compounds in particular. Yeah. So we don't have any more questions from the attendees. If you want to add something or to conclude.
I think we got in the chat a paper on the fish intake and the gut microbiota, so that could answer the question.
From Oddburn, Anita, thank you. So for the, really nice. Thank you.
No, but we really thanks a lot, Rachel, for putting this up. I think that's interesting, and of course we have this network. We are non-commercial and we are academic. So, but of course, if we evolve, there might also be perhaps some interaction with the industry. But that will probably be the next level. So our focus is really to put this into clinical perspective, but I see that this is an interesting field for several actors. So I think we can always be open for dialogue. So and exchange ideas, I think is always very fruitful.
Actually, it's really a huge trend now for the food complement in general to look at the microbiota. It's a new trend, and but as you say, and what we can sum up from this webinar is that we just scratch a little bit on the microbiota studies, and we need more documentation. And this is also the role of the project Marine for Health, is to support this documentation, because it can take time to develop a new product and to put it on the market. So it's always nice to have academical, to have you guys, scientific, to support the industry. And I want to say thank you for your participation. It was really interesting, and a lot of information and a lot of studies and projects. So it was really nice.